I am Benjamin. I'm at the Australian National University, and we normally work on plant pathogenic fungi with a focus on rust. And we do also some other stuff, but that's kind of our main thing. So I'm the host for the seminar today. So a brief intro to the series. This series is open to anyone. We had last week our first seminar by Dr. Susan Auer talking about sclerotic disease. You can find her talk on the Open Plant Pathology website, including the full video and the slide deck. And maybe Adam can put this link into Slack again. So some housekeeping. You can ask questions in the Q&A button. So the Q&A button is at the bottom of the Zoom interface, and you can post the question. And I will try to summarize them at the end of the talk and present them to Remco. If there's more questions than we can answer during the time, you are welcome to join the Slack, and there will be an opportunity to interact with Remco potentially afterwards and ask him some more questions over the coming talks. The channel is virtual seminars. Also, the talk will be posted on OSF and including the slides, which will be the UI, so you can also cite it, and it becomes discoverable. All right, with no further ado, we're going to hand over soon to Remco Stam. Dr. Remco Stam is a MPI at Technische University of Munich, Technische Universität München. And he will present his talk about the diversity and molecular evolution of plant defense against pathogens in nature. Thank you very much for Remco for taking the time to present to us today, and we're looking forward to your talk.
Yeah, thank you, Benjamin, and also thank you, Adam. Good morning, everybody from Germany, from my side. So as Benjamin said, my name is Remco Stam. You can follow me on Twitter, EvoMPMI. And today I would like to present some of my work on diversity and molecular evolution on pathogen resistance in Solanium chilensis. And I'll start with some very general introductions, right? So I think most of the people that are watching this video are familiar with this. So I want to know what factors are involved in evolution in a natural situation and to what extent. And to understand this, we need to understand the models that are out there. So we know that there's these several layers of defense responses in plants. So we have this PAM-triggered immunity or pattern-triggered immunity with pattern recognition receptors that lead to a signaling pathway that trigger immune responses. And we have this other layer where secreted proteins or effectors of pathogens are recognized by R proteins, coded by NLR genes, and that also lead to some kind of immune response. We know that these things interact, right? Effectors can prevent PTI from happening, and we know that there is actually a hell of a lot more out there that also interacts with the pathogen. There are physical barriers, there are preformed metabolites that stop the growth, there are toxins, the proteases, everything's out there. There's a lot of crosstalk in between all these things, and there are a bunch of induced secondary metabolites. So what I really want to understand is, what is the genetic diversity involved in maintaining the resistance in natural populations, and which of these factors play a role to which extent for any of them, right? So there are a lot of things here that interact, and it's not just one R gene or one PRR that is doing this. But you got to start somewhere. So I'm actually going to present work on studying NLR genes that code for R proteins. That's the main thing of my presentation today. But I will branch out. I will also show you what I'm going to work on in the next couple of years. So we're talking about immune responses and how we measure them. And I'll give you a very brief sneak preview on what we hope to be doing with effectors as well. So on the pathogen side. Right. So to study what happens in nature, you need a system. And I think Solanium chilensis is an excellent model system to study diversity and evolution of pathogens in a natural situation. So Solanium chilensis grows in a relatively limited range. So we can see it here. It grows from the south of Peru to northern Chile. And before I started all this work, other people have already defined that there are basically 4 genetic groups. So we have a northern group, a central group, a southern highland group, and a southern coastal group. And that's it. There's not more further south, there's not more further north. There are other wild tomato species, but that's it. Also important to note, I'm moving my cursor around here, this is the Atacama Desert. So it separates the coast and the highland populations. which means that even though geographically close, they're actually furthest apart because they're split by a desert, so they cannot really cross-fertilize or interact with each other. Right. And all these populations, so each dot on this map is a population, and all these populations grow in their own unique habitats, right? And this means that all these populations have adapted to microclimates, to very specific environmental changes, but also very specific biotic stresses. At least that's what we hypothesized. Just to give you an impression, these are some of the sites that we visited when we were looking for these plants in nature. So we have here on the southern highlands areas, we have some plants that really grow in deep gorges, or they grow just outside of them. So it's very sheltered and very specific, because the rest, if you look at the horizon here, it's all desert, so there's really not much. But in these gorges, there's water. Then on the lowland side, we actually it's completely barren, but there's still runoffs, there's still rivers. You can see here the cracks in the soil where the water has run, and there's a very tiny plant down here. If we go to the northern part of the range, so the central region on the border between Peru and Chile, we actually see that the climate is quite different. So we still have these dry riverbeds where there is underground water flowing, but we also have much wetter climates, much more rainfall if we go higher in the mountains. So we really expected that this would lead to a very strong diversification also in terms of pathogen pressure. And to test this, we basically went along. We took a couple of pathogens just from our lab, like an Alternaria strain that we found on potato, which we know also grows on tomato, Fusarium, Phytophthora, and we tested different populations. So we took a set of 10 plants with from a certain area, and we took 7 different populations, 10 plants per populations, and just infected them. And what you can see is a range of symptoms. I'm not going to show all the details. Everything's been published a couple of years ago. But important to notice that in some cases, plants were completely healthy, no infectious symptoms at all, or maybe, as you can see here, very neat little hypersensitive responses, so successful defense responses. And in some situations, plants were infected, mildly infected or fully infected. So we have a plant system here that has a geographical spread, and it has very clear differences in resistances. We also have a very nice reference genome. It's very fragmented, but we have very good protein models predicted, and we're working on actually scaffolding everything together now. And we also managed to have a good NLR annotation, and this is of course important if we want to study evolution of resistance or NLR genes, these resistance genes. So we found about 230 NLRs, and all the clades that have been annotated in other selenaceous species, they are actually there. So we find this TNL clade, we find the RPWA clade, we find the NRCs, which are the so-called helper NLRs that are kind of dimerizing with the guys on this side of the phylogenetic tree in order to actually make these guys function. And we have this group here that actually functions on their own or as homodimers. So we have all of them, and the numbers seem to match. We found 236, and in Solanum pinelli, you find 216, and in some other species, you find about 250. So we're pretty sure we have a good reference with good annotations here. And another thing that we have for Solanum chilense, and it's linked to what I showed before, this nice species distribution, is that we know the demographic history. And this is very important, because if you want to study evolution of a species or evolution of a gene family, you need to know how the species itself evolved. So we know now that the central region was the origin, or maybe the north, but we take the center as the origin, and then the different subpopulation split. So the southern population split of the central population about 30,000 years ago, and the populations on the coast separated a lot earlier. They separated about 200,000 years ago. If you're interested in this kind of analysis, please check out the paper. This is work done by a very talented postdoc in the population genetics department at the TU in Munich. And all the details are all there in the supplement. So everything in terms of reproducibility and looking at code, we have it all there. So it's really worth looking into. So we have a great model system now. We know how it originated. We know we have a reference genome, and we know we have phenotypic diversity and resistance. So we can start testing whether these NLR genes, these typical resistance resistance protein coding genes, are evolving in the species, and how they are evolving. Are all of them evolving? Is one of them under strong positive selection pressure? What is happening? So to do this, we take, again, a set of populations. So we have 14 different populations from all the different regions, including some that we used to calculate the demography. And then we take 14 populations, 10 plants per populations. We do this targeted enrichment sequencing, some people call it RenSeq, where we sequence the NLRs. We do this in pools to save on money, and then we map back to a reference. We do a lot of quality control because we had MySeq reads, so we have relatively short reads, which can be problematic for these NLR genes because they're quite repetitive. But we did a lot of controls, and all these controls and tests you can see in our genome biology and evolution paper. So we're absolutely sure that for those genes that we're calling the SNPs, we have accurate data. We really filter out a lot because what we really want to see is whether this sequence evolution is something that is really happening in NLRs. So copy number variation is something completely different, and we cannot assess with this method. But sequence evolution, positive selection, balancing selection, this is something we can test. So we call the SNPs, and then you calculate the evolutionary parameters. And there are a lot of things that you can calculate. And this is just a very short list, actually, of the different parameters that you can calculate. And what is important, and we tested a lot of them, we tested much more than this, is that with the kind of data that we have, we can reliably assess nucleotide diversity and also the ratio of the polymorphisms in the genome. We can, of course, calculate nucleotide diversity, but then for synonymous and non-synonymous mutations, right? So a mutation can be a mutation and the protein sequence doesn't change, or a mutation can actually make the protein sequence change. When this happens, when it changes the protein sequence, this is an indication of diversifying selection or diversification or maybe balancing selection. So these are the two main statistics that we can robustly calculate, and there's another one called FST, which I will highlight later. Let's first just have a look at some of the results. So what do you get when you sequence 140 plants, all the R genes, and then you map the nucleotide, you calculate the nucleotide diversity for each of the genes? You get a plot like this. I should say we also calculated the statistics for a set of control genes, we call them CT, just to see what's happening in a set of normal housekeeping genes, like development-associated genes. And what we see in these development genes is exactly what we expect. We expect, first of all, a very low nucleotide diversity. So, and the nucleotide diversity is lowest on the coast and in the south. This makes sense. These are the smallest groups of populations. The smaller your population is, the lower your diversity is. This is a given. So then the center is a bit bigger, a bit higher, and in the north we see a bit more diversity, probably because there might be a bit of mixing with another species called Solanum peruvianum. So we don't really pay too much attention to this. If we then look at our NLR genes, we actually see that the nucleotide diversity on average isn't that much higher, right? We see that the coast is still as low, the south is a bit higher, and the center and the north are also not that much higher if you look at the averages with these box plots. But what we do see is that there are a large number of outliers. So indeed, there are some NLRs that really have a lot more polymorphisms in this data set than you would expect in a set of control genes. Now, the other thing that we can check is, are these mutations more often non-synonymous mutations? And that's what we see here. So for our control genes, we see that actually most often they're not. We're well below 1. This is the ratio, right? So at 0.1, there are 9 non-synonymous versus 1 synonymous mutation, so to say. Not quite, because it's not how you calculate, but still. And for the NLR genes, we see that the value is still below 1. So it's still not that much diversification going on, but it's a bit higher. And what is very cool is that we really have this cloud here of NLRs that really seem to have a lot more non-synonymous mutations than synonymous mutations. So this is interesting, right? But we want to understand a bit more what is happening. So you can take these data. Here, each dot is one gene in one population, right? You can take this data and summarize it in many different ways. So one way to summarize it, for example, is looking at these clades that I described earlier. And this is important because this NRC clade was hypothesized by the lab of Sophie Kamoun to be very conserved because it's kind of an integral part in the signaling network. And this is one thing that we see. If we look overall, we see indeed that this NRC clade is amongst the most conserved. It has at least the least non-synonymous mutations, or amongst the least non-synonymous mutations. And what's also interesting is that, for example, the CNL21 or CNL20, these are clades that we newly describe in Solanum chilensis. They do not occur in other species. And these are the most diverse. So evolutionary, that makes sense. So you have your helpers that are definitely amongst the most conserved side, and you have your sensors that are more diverse. And the newer sensors that are new in this species are even more diverse. But this is still a bit too generic maybe. And what we really want to see also, we wondered, can we see if there are specific genes that are really under strong positive selection in one population in the north, or under purifying selection somewhere else? Well, you can also plot the same data. This is all still the same data, just split in different ways. So talking about R and reproducibility, there are a lot of scripts here that you could dive into basically. What we see here is we have all the individual genes that we were able to test on the x-axis, and then the pi n pi s value again here on the y-axis. What we see is, if you look at the genes, there's not really one gene that is an outlier there. It seems to be that there's some here that are a bit higher, but they don't really belong to known clades. These are either non-determined or new genes that we cannot really work with. And we see that the outliers in generally tend to be more dependent on which population we sample which gene in, right? Sometimes you see here green dots, and in this case, a green dot means that we sampled this value in a population on the coast. Here is a blue dot, here is a purple dot, here are some red. So outliers are there, but it's every gene is an outlier in a different population. So to work with this, we need to do another calculation. So we need to go back and we need to start comparing what happens between the populations. So this is where FST comes in, and it's a relatively simple calculation, and it gives you a number from 0 to 1, where 1 means complete differentiation, 2 different alleles when you compare the populations, and 0 means that the mutations are shared. So this is what we can calculate for all the genes, for all the populations, every pairwise comparison. So you get 91 pairwise comparisons for 91 genes, which gives you, again, a lot of dots on a plot. And yeah, then we still have something that looks like a kind of even distribution. There's not a single outlier. And you see, again, on the x-axis, you see all the different genes. And you see that in some cases, the values are quite low. In some cases, for a certain gene, they are a bit higher. But what does this mean? How do you define an outlier? And moreover, we know that FST is higher when species are further away from each other. I mean, if you're geographically distant, you're more likely to be differentiated. So your FST is going to be higher. I look more similar to a German person than I look to someone from China, for example. It's just geographical distance. This is how evolution works. And what you can see is also in our populations, we see this. So everything that is gray is within region comparison, whereas, for example, the dark blue compares the north and the south. And you see the north and the south is always higher. So this still doesn't tell us anything on which genes are really under selection or changing selective pressure. And this is where the demography comes in. So we have this model, we know when the population split, and this also means that we can simulate what happens when you have a neutral evolutionary scenario, right? Just a species going from the north, splitting on the south, on the coast, and in the mountains. That generates diversity. We can calculate this, we can estimate this, and then we can compare this estimation with our actual values. And this is what we've done. So again, for each comparison, we can show what we expect. So this is a simulation, the absolute extreme values of our simulation. So when we compare two populations in the south, we expect FST to be roughly 0.25 with a couple of outliers. If we simulate 30,000 genes, so a full genome, the absolute outlier is here, about 0.6. What we then see in our NLR data is that there are definitely some genes that are outside. So these genes are the genes that are under strong evolutionary pressure changes. And we see this for almost every pairwise comparison that we do. So if we compare a population on the coast with a population in the central region, we also see this expected range. It gets a bit bigger because the populations are further apart, but we do find outliers. And here again, we can do this for all 91 comparisons. In most cases, we can find 5 up to 7 outlier NLRs. Interestingly, these are not always the same NLRs. This dot here might not be the same NLR as that one there. So the question that we now have is, which R genes are the ones that are really evolving between the different populations and which ones are the unique ones, right? So we can map for each individual NLR how often we find it as an outlier. We can draw this as arrows. So some genes we did not find at all. There we go. But we find 53 NLRs that each show very specific patterns as outliers. So this one, nothing. This one we found, but only in 2 out of the 91 comparisons. So we think, and this is robust, right? We have 30,000 simulations. We take the extreme outer values as a cutoff, not even a 5% or whatever. So we know that this is truly differentiation. But we still want to be on the safe side, so we think this is more kind of a local adaptation. These 2 plants, our gene is different in these plants and these populations, but not in all the others, right? But there are also genes where we see that really the arrows point all over the place. So this one, and also this one, if it's coming now, yeah. So, and these, where we really see these changes between all the populations, or most of the populations, compared to one other population, we think these must be the main habitat adaptation NLRs. So these are the NLRs that are kind of driving the adaptation or helping the species to really adapt to something new. Remember, the species started in the north, migrated south, and when you migrate south, you encounter a new biotic environment and then a new stress environment and new pathogens. And we think that these main habitat adaptation NLRs that we pick up all over, these are the essential ones that really help the plant species cope with the new stresses that they encounter. And the interesting thing is, that's really not so many of these genes. There's only 4 when we go from the center to the coast, only 4 when we go from the center to the southern highlands. There are a few more when we go up north, but overall, there are really not so many. But what is interesting is that when we look at the distribution, which classes they belong to, is that they really have a very different distribution from the classes that they belong to than the local ones, so all the others that are not so general. So if we look at our local adaptation NLRs, we see that they're mostly not belonging to a clade, or they're actually these sensor NLRs. Whereas if we look at the main habitat adaptation NLRs, they're much more often TNL, and also, and this was a big surprise to us, much more often belonging to these NRC. So they're much more often belonging to these helper NLRs. And I think this is very important to realize, that if you have a network of R genes that is interacting, that is working together, and you need to migrate into a new area in a new region, you need to make sure that your whole network stays robust and adapts. So this is probably why these helper NLRs are actually adapting also. So on average, they're really conserved, but when they change, and there's only a few of them, then we see that they really adapt all over the species. So by studying these NLRs, we learn actually quite a lot of cool things of how this variation and diversity happens in nature. So we really have clear proof of sequence evolution. Many NLRs show a lot of variation, but definitely not all of them. So it's not the case that all the NLRs are evolving all over the place all the time, but the non-synonymous diversity is definitely higher than the normal for all genes. So the different clades really show different rates of adaptation. And this also means that if you look, and if you want to look for something specific, you definitely have new sources of R genes here, because our NLRs really locally adapt to biotic stresses. But there are only very few genes that show signs for these changes throughout the whole species. Very few major genes. And surprisingly, the NRC are overrepresented in these major genes, which gives us again a cool evolutionary result, that the network position, being central in a network, doesn't mean that you cannot evolve, which is a very important finding actually. It means that you don't have to be conserved, but the network probably needs to evolve with you. So this is really cool. So this is the part on R gene evolution, and I think we learn a lot from this. But there's something that was kind of bothering when we were working on this, and this is that when we go out in the field, we don't really see this typical R gene black and white resistance. We always see that there is some kind of quantitative resistance in nature. And this is why in my new project that started about 1.5 years, almost 2 years ago, we're really looking into what are the quantitative differences for resistance in Solanum chilense. So I'm going to give you a little bit of a preview of what's going on in the lab in the next 5 minutes. So we have very clear differences in quantitative resistances between the populations, I showed you already, and also between the plants. So this is work by Winter alone. And here we tested 10 plants from one population, samples from the center of our species range, and we really see very reproducibly and robustly that there is this variation. Some plants are definitely more resistant than others, but all of them are a bit susceptible, right? All of them can sometimes get infected. You can see this also here at the pictures at the top. We have one plant that is always 100% infected. That's our most susceptible plant. We also have one plant that is, on average, only 50% of the lesions somehow develop and they never really grow. So this is a very clear distinction within one population. So within one accession number, within one geographical area. This is important to keep in mind. And it doesn't matter where we test the plants. We see this everywhere, right? Also, when we take a population from the south, we see, again, a lot of variation. This population, on average, is a lot more susceptible, probably because the pathogen pressure in this area is different, is lower for this specific pathogen. We tested with Phytophthora infestis in this case, might not occur there. But we still see this variation, and this is extremely consistent. We do a lot of infection assays, and we can really, really repeat this very well. So now the question is, what is really causing this? And this is where we're starting large-scale screening with elicitors and really trying to measure different defense responses. And I'm not going to show all of them. I'll just show you one graph just to give you a bit of an idea. And I'm happy to discuss what this means and how we can build on this in the future. So I take this population where we have our most susceptible and most resistant plant here. The difference between the two of them is not that big, but maybe we see something in terms of defense responses. And one of the defense responses that people very often look at is, is the reactive oxygen species burst. So plants make reactive oxygen species directly after they encounter a pathogen. And this is what we see here. So we measure this in all these plants, and we actually see that our two most susceptible and most resistant plants, they're very close together. But there are some plants that have a lot more reactive oxygen species made, and there are also some plants that don't have anything made. So this, we now know, and this came a bit as a surprise to us actually, that ROS burst in Arabidopsis is often seen as a proxy for if you have high ROS, you have a very resistant plant. Here we have one plant that has absolutely no ROS burst at all, yet it is still as resistant as some of the others. And we have some plants that have a very high production of reactive oxygen species, and they're definitely not more resistant or more susceptible than the others. So we're now really building on this. We're trying to work on screening more populations, screen more different responses, so we can also screen hormones, right? Ethylene, salicylic acid, different metabolites, and we also want to check what happens when we screen with different elicitors. We're also at the same time working on getting more genomic information, so we've resequenced about 40 plants at the moment, and we want to ultimately get this all together in a model of defense diversity in Solanum chilensis. So if you have some suggestions of other things that we can measure in a larger scale, I'd love to hear them, because it's a lot of work to sequence these, to measure all these different responses in these plants, right? So this will be the next step in our research, and what is something that we really want to add onto this is not just checking at what is the generic defense response. We're now screening with an elicitor, with a derived molecule that represents the pathogen cell wall, but we want to ultimately also do the same experiments with locally obtained pathogens. So pathogens that we found in the field on the certain populations, on certain plants, and then really see if those plants where we found the pathogen are more or less resistant to the others. So in the last 2 years, we've had a couple of field trips in Lima. So starting from Lima and then going south to Tacna, Antofagasta, all the different areas where we have Solanum chilensis growing, and a couple where we have other wild tomatoes, just to make sure that we can make a good comparison. So with 76 sites and about 1,500 leaves, we now have about 516 purified samples, and most of them seem to be Alternaria alternata forma specialis. And in the next couple of months, we hope to be really working on a pipeline to sequence them and really analyze how different they are and how they behave on the different host plants and the different pathogen populations. So this is a great collaborative effort, especially with a lot of people also in Chile and Peru. Without their help, you cannot do any of these things. So yeah, I hope you enjoyed this. I think this was a bit of an overview of what I've been doing. So I think very important is that we can really show NLR evolution. So we can really show on a shorter time frame within one species that NLRs are evolving. There is clear proof of sequence evolution. NLRs adapt locally to biotic stresses, but we also see these species-wide trends, maybe to adapt to climatic changes. And definitely we see that because the NRCs are overrepresented as major genes, that their position in the network doesn't mean that they're not able to evolve. And then on defense responses, we think we can open a whole new line of research trying to understand all this variation and all this diversity. And hopefully, when we get the pathogens from these areas isolated and sequenced, we can actually work with them and really put everything together and really try to understand how coevolution happens in one species on these different population levels. So with that, I'm at the end of my presentation. I thank everybody very much for your attention, and I need to thank all these people for their help in the lab and all these people for funding as well. So thank you very much. Oh, beautiful, Ben, beautiful.
Right. Thank you. Obviously, your crowd is very enthused about your talk. So we had a couple of questions, and so if other people still have questions, please feel free to put them in the Q&A box. So I start with one, and this was about, so it's the first part of your talk. You talked a lot about the diversity of NLRs and NLRs which involve faster, and you kind of focused on the stuff which sets the populations apart. But what do you think about why do actually some NLRs don't evolve, or why are they? Do you find also some which are super conserved, and what could be the function of those?
Well, that's a good question, right? And I think there might actually be some that are essential. So there might be some that experience purifying selection. The problem with the system as we have it is that we don't know the function for basically any of them. I mean, we know we have a homologue of known R genes, but we don't know the function. And this is also why I moved away from this R gene study and move into something where we first see phenotypes. But I think it's very well possible that some of them are under very strong purifying selection. You need this R gene in this form to be able to detect the pathogen in that particular population, that particular area. So having a certain, if the pathogen has a certain effector that is very essential, you need an R gene that always recognizes this one. Might be the case, but it's really hard to prove these things without knowing what pathogens are out there. So this maybe in the future we can revisit this kind of questions again. Yeah.
Cool, thank you. So another question. So from Ella. So the person, sorry, wants some resources on how to study and understand the basics of evolutionary biology. Maybe you can answer this later in a Slack. Might be good. The other question, which is also kind of related, is what do you think is the importance of bioinformatics in studying this plant evolutionary biology?
Well, I guess what is very important is that you know where your species come from, how they relate. So I think this is the strong point of our system. We can look at different populations. We know which one is most likely being the ancestral one, and then you can really start saying something not just about diversity, but really about evolution. And I think in any kind of system, this is what you need. You need to be able to compare what is normal. I mean, nature doesn't stand still. Nature always evolves, right? There's always something changing. So to really have a system where you can separate normal changes just by chance from changes because of strong evolutionary pressure, this is what you need to get before you can start answering questions. And I think this is, for example, no offense to anyone working on it, but this is something that is very hard to do in Arabidopsis, for example, because the demography of the species is very unclear. It's very rapidly spread by humans, so you really cannot do the same kind of analysis that we can do here. So you can find outliers, but you cannot say which one came first and which came where, and etc. So to really understand evolution, you need a system where you know where your species came from, how it developed, how big your populations are, etc. And this is sometimes really hard to get, especially in a world where humans have moved plants and crops and everything around so much. So
cool, thank you. So another question was, so do you like what's the role of transcription factors in your system? Could you look at this?
I cannot look at this at all. So in this system, so in the R gene part, I have absolutely no idea about the transcription factors, right? It might be that there is still a whole new layer of complexity. In this second part, where I'm starting to test the defense responses, there definitely might be something related to transcription. So we're actually, for these plants that show a very high growth burst, we also measured several hormones in those. So hormone responses, ethylene, salicylic acid. and we see that the transcription is already higher for some of the genes that are a bit further down upstream in the pathways. So there seems to be definitely a transcriptional regulation in there. So it might be that we need to start looking at how these transcription factors are diverse rather than the actual genes in the pathways. But this is actually the next step of the process. Is it now the ROS pathway itself that is affected, or is it the regulation of the ROS pathway that is infected? And how does that then relate in the end to the resistance that we observe in the plants, or to the responses that we measure in the plants?
Cool, thank you. There was a follow-on question to your answer, but you just had about Arabidopsis. And the question was from Lisa Rothman. Does it mean that the Arabidopsis outliers are too random?
Oh, yeah, I don't know. I'm afraid to answer this. I don't know. So I know in Arabidopsis you have this so-called relic group, like a group of accessions from Spain. And I think pretty much if you compare those with some of the accessions that you find in Africa, or some of the accessions maybe also in Sweden, you might find something that is useful. But if you compare, I don't know, an accession that was sampled in the north of Germany with an accession that was sampled in the south of Germany, it doesn't mean that this is where they originated from, because they've been sampled like, what is it, less than 2 decades, 3 decades ago probably for some of them. And the seeds move, they move along train tracks, they move back and they move forward. And so, yeah, it's very difficult, but you should be able to find some more ancient accessions. I think Angela Hancock at the MPI works more in Africa, and it seems to be more untouched, and you really see these more clearer demographic patterns in there, for example. So I don't know. I'm not an expert in Arabidopsis. I just know that it's really hard to assess the demography in a way that you can do for certain other species much easier. So, yeah.
Cool, thank you. So then from Ralph Frickelhofen, I think that's your boss. Does helper evolution really represent network adaptation, or could alternatively the helper be direct affected targets for affected trigger suppression of ETI?
I think he answered the question himself already. I think we don't know. I mean, so it could be directly targeted and therefore directly under selective pressure to evade recognition. This is very well possible. I think it might not be, but I don't actually know why I think this. I think I have this hypothesis in my mind that it might be more something of temperature regulation. So most NLRs are temperature dependent. And if you migrate south, the daytime temperature gets higher. So maybe these NRCs are really just evolving to be more stable when it's hot, for example. This would be a very simplistic explanation from my side. But that's why I like it. But it might also be that they're actually in the south, they're more targeted by a certain effector from pathogen Y directly. So yeah, something to test as well, but cool. I don't know how.
So there's 4 more questions, if you have a little bit more time. So Shyam, there's maybe more. Shyam Solanki, he is asking, did most of the NLRs in the study, okay, so where are your diversifying selection happening? Like what residues are actually mutated? Are they in the LLR region or the MBS region, or within the protein at the end? Like what has actually changed?
So we didn't look too much into the details here, but a quick look showed that it's all over. Mm-hmm. So in our paper, we did the first paper on this kind of method we did in Solanum pinellii, where we really looked specifically in those genes that had diversity, and there we saw it doesn't matter. In some cases, you see a few mutations in the MBS that are definitely under changing selection, in some cases NLRs, in some cases in the CC domain. For Solanum chilense, because we have so many genes and so many patterns, we kept it more general. So we didn't really try to summarize this, but it is all over the place. Yeah.
Okay, so there's a question from Kahiti Mata, which is more like a methods question, and then maybe just pass this on to you. You can maybe dig into this a little deeper later on, so I'm going to skip this for now. Because I think it needs a longer explanation. And then the other question from Diana Ramirez is, you talked about the absence of resistance in plants that are able to produce ROS upon HAMs. Could you elaborate on this a little bit more?
So the graph
And
that I
are plants
showed
unable to? Yeah, go ahead.
So the graph that I showed that some plants do not produce any reactive oxygen species upon elicitation with an elicitor that we use, like laminarin, which is a proxy for the Phytophthora cell wall. We don't know what this means. So to me, it was a big surprise because everybody says you need ROS to trigger your defense responses. We didn't see this. It might mean that these plants, because they're not less resistant than some others, it might mean that these plants recognize another PAMP very well and that therefore they're still equally resistant. It might mean that they have other compensatory mechanisms. They might have a thicker cuticle. They might have, I don't know. This is exactly the kind of answers that we're looking for. So what could be the other mechanisms that keep everything in balance? And why is it that this plant is not completely dead and completely attacked by pathogens, and that it still exists in the population? So I hope that answers.
So the following question was, could this be linked to the oxidative cellular status depending on temperature or range?
Probably, but I'm not enough of a plant physiologist to answer this question. So these are the things we're really just exploring. So we're really, anyone with more plant physiology experience than me is happy to tip in and give help hypothesis and think of how we can bring this project further together, because we're really seeing things that we never expected, so much variation, so many different things, and it's so hard to measure everything in all these plants in a streamlined fashion. So, yeah.
Cool. All right. It's nearly all the questions done except for mine. So the question I had was still, so you measure these quantitative traits, and I guess you infect all these plants in the same environment. So you put them in a growth chamber and you have the same environment. Do you think if that environment can interact with your defense responses depending on the range where you actually sample them? So if you would mimic the environment that's in real time on, they would actually respond differently?
Yes, so this will be my next big thing if someone is willing to support and fund and work with this. So what we want to do is now we want to take the pathogens that we found in Chile and infect them on plants from the different areas in different climatic regimes and see how this affects their the way they infect, and also how this affects the defense responses that we observe. Because everything, like you said, everything is done now. It's detached leaves in a box. So it is very much simplified, but the fact stays that we see differences in these defense responses. But to really understand them, we need like climatic modeling chambers where we really can emulate the difference between a plant growing at 3,500 meters altitude in a very humid environment versus a plant growing on the coast where it's bone dry. You need to be able to take this into account to understand what's happening. So yeah.
Cool, that's really cool. Okay, I have one more question for you. So how are you going to do all this stuff now with the travel bans?
Sorry, with the travel bans?
How are you going to go to Peru?
Well, the good thing is we already have 500 isolates, so we don't have to go to Peru at the moment. So we have a lot of things that we can test in the next year. And hopefully stuff lifts in the next year and we can actually go out there. But that said, like I mentioned, a wonderful set of collaborators in Peru, and they worked with me in the field, and they can also work without me in the field. And this is essential. You need people that know the fields, that are able to also help there. We cannot take pathogens or leaves out of Peru without permission. We cannot take anything out of Chile that has plant DNA on it. So we need to collaborate with people there, and they're really eager to help purify the pathogens, get the right permissions. So I think we can also work without going there all the time. It will be slower, it will be harder, but it should work. Yeah.
Thank you very much. That was a very exciting talk. And also thank you so much for staying on and for this exciting discussion afterwards. And I think it will be also useful for people afterwards to listen back and maybe also learn some more about your system. And I'm really excited to see where you will go in the next couple of years, because I think you established quite some very exciting resources to matching up like the pathogen diversity with the host diversity. Really cool stuff. Thank you for your time. All right, so we're nearly at the end. Thank you, Remco, for your time. So we have a little bit of an Easter break, so please stay at home and don't spread the germs. In the meantime, we have another talk in 2 weeks' time, again on Monday, roughly the same time. April the 22nd, and this time it will be Dr. Peter Solomon from Professor Dr. Peter Solomon from the Australian National University, and he will talk about how the study of necrotrophic effectors advanced our understanding of the enigmatic PR1 protein in wheat and other systems. So please come back in 2 weeks' time and join us last moment to thank Remco for an excellent talk. Cool. Thank you. See you next time.